All right, let's talk about something that's happening inside you right now, a crucial detox process that, let's be honest, is a big deal for your exams. We're going to master the urea cycle. Let's dive in.
So think about this for a second. Every time you eat protein, your body breaks it down and creates a byproduct, ammonia. And this stuff is, well, it's really, really toxic, especially for your brain. So how do we deal with this constant threat, day in and day out, without even thinking about it? Well, it turns out our bodies have a pretty brilliant solution. And that solution is the urea cycle. You might also hear it called the Krebs-Henselite cycle. This is the body's main game plan for detox. It all happens mostly in the liver. The whole point is to grab that dangerous ammonia and convert it into something called urea. You can think of urea as a safe, water-soluble package that your kidneys can just flush right out. Pretty smart, right?
Okay, so here's our game plan to make sure you've got this down cold for your exams. First, we'll talk about why ammonia is such a problem. Then we'll see where this all happens inside the liver cell. We'll walk through the 5 main steps, cover the costs and controls, see what happens when the cycle breaks, and then we'll wrap it all up with a quick recap. Let's do it. All right, section 1. Let's start with the villain of our story, ammonia. Where does this stuff even come from? And why is it such a threat?
So where's all this ammonia coming from? Well, it's not just one thing. The number one source, by far, is the metabolism of amino acids, the building blocks of protein. But we also get it from breaking down other things like purines and pyrimidines, and even from the bacteria hanging out in your gut. The bottom line is our bodies are making this stuff all the time. It's just a fact of life.
And I really want to stress this. The stakes here are incredibly high. We're not talking about a mild toxin. When ammonia builds up in your blood, a condition we call hyperammonemia, it's a full-blown medical emergency. It's poison for the brain. It messes with energy production and can cause some really scary symptoms, which is exactly why the urea cycle is so absolutely critical. Okay, so we know our body has to fight this ammonia battle, and the battlefield is the liver. But here's where it gets really interesting. It doesn't all happen in one spot within the liver cell. It's a story of 2 different locations. This is a super important point, so definitely file this away for exams. The urea cycle is split between 2 different parts of the cell. Think of it like a relay race. The first 2 steps happen inside the mitochondria, the cell's power plant. Then the baton gets passed, and a molecule moves out into the main cell fluid, the cytosol, for the last 3 steps. This whole mitochondria then cytosol thing is a classic question, so make sure you remember it. Okay, let's get to the heart of it. We're going to walk through the 5 key steps of the Krebs-Henselite cycle. This is the play-by-play of how ammonia gets neutralized. All right, we're starting inside the mitochondria. Step 1 is the big one. It's the rate-limiting step, meaning it sets the pace for the whole cycle. Here, an ammonia ion and carbon dioxide get fused together to make something called carbonyl phosphate. The enzyme doing the work is carbonyl phosphate synthetase I, or CPSI for short. And this step isn't free. It costs 2 ATP. Then, for step 2, that carbonyl phosphate hooks up with another molecule, ornithine, to make citrulline. And here's that handoff we were talking about. Citrulline is our shuttle bus. It's the molecule that gets actively pumped out of the mitochondria, carrying that nitrogen atom with it, and into the cytosol. The race isn't over yet. The final laps are about to begin.
Okay, we're in the cytosol now. Step 3 is where we bring in the second nitrogen atom needed for urea. And where does it come from? It's donated by an amino acid called aspartate. Aspartate and citrulline combine to form argininosuccinate. Next, in step 4, that molecule gets split in 2. We get arginine, which stays in the cycle, and something called fumarate. We'll come back to that one. And finally, the grand finale, step 5. The enzyme arginase chops the arginine, releasing our end product, urea, and, crucially, regenerating ornithine.
And this is what makes it a cycle. That ornithine molecule we just remade, it's now ready to go. It gets transported back into the mitochondria to pick up another molecule of carbonyl phosphate and start the whole process over again. It's this constant recycling that makes the system so incredibly efficient. So, we've gone through the steps one by one. But now, let's zoom out a bit and look at the bigger picture. We need to talk about how much energy this costs, how it connects to other things happening in the cell, and how our body keeps it all under control. First up, the energy bill. Detoxing ammonia isn't cheap. To make just 1 molecule of urea, the cell has to spend 4 high-energy phosphate bonds. Now, you might notice that comes from 3 ATP molecules. The reason is that one of those ATPs is broken down in a way that's like spending 2. So just remember the final tally. One urea molecule costs the cell 4 high-energy bonds.
Now remember that fumarate we made back in step 4? I told you we'd come back to it. Well, it's not just thrown away. Fumarate is a key player in the TCA cycle, or the Krebs cycle. You know, the central hub of energy production. This is such an elegant connection. The body links its detox pathway right into its energy pathway. It's a perfect example of how nothing in metabolism happens in isolation.
So how does the body control this whole process? Well, it all comes down to that first enzyme, CPSI. That's our main control point. It gets switched on by a special activator molecule called N-acetylglutamate, or NAG. Think of NAG as a gas pedal for the enzyme. When is the pedal pushed? Well, after you eat a big steak or any protein-rich meal, your body makes more NAG. This fires up CPSI, which speeds up the whole urea cycle to deal with all the extra ammonia. Simple as that.
Okay, so this pathway is obviously super important. So what happens when it breaks? As you can imagine, if any step in this assembly line fails, the consequences can be really, really serious. Let's touch on the clinical side of things. If you have a genetic defect in any one of the 5 enzymes we talked about, it causes a specific urea cycle disorder. You can see them listed here. Each disease corresponds to a specific broken enzyme. And even though they all have different names, they lead to the same fundamental, dangerous problem.
And that problem is hyperammonemia. The detox pathway is blocked, so toxic ammonia backs up in the bloodstream. And as we've said, that ammonia is poison to the brain, causing all those severe neurological issues like brain damage and coma. It really hammers home just how non-negotiable this cycle is for our survival.
Whew, okay, that was a lot. But we're almost there. Let's bring it all home and do a quick final revision to lock in the most important points for your exams.
All right, here are your absolute must-know takeaways. Ready? Number 1, its job is to detoxify ammonia, mainly in the liver. 2, it happens in 2 places, mitochondria and cytosol. 3, it costs 4 high-energy bonds. 4, it's linked to the TCA cycle through fumarate. And 5, if it breaks, you get life-threatening hyperammonemia. That's the core of it. And of course, trying to remember all those intermediates in order can be tough. So here's a classic mnemonic to help you out. Ordinary, careless Albert always argues. That's for ornithine, carbamyl phosphate, citrulline, arginosuccinate, and arginine. Trust me, it can be a lifesaver in an exam.
Okay, one last pop quiz to make sure you've got it. Which enzyme is the main control point of the urea cycle, the one activated by N-acetylglutamate? Think about it for a second. It's carbamyophosphate synthetase I, right? The enzyme for that very first step. Awesome job. You've just walked through one of the most critical pathways in all of biochemistry. Keep this fresh in your mind and you're going to do great.